నాకంత అదృష్టం లేదు కానీ ఇంకా ఆపిన వీడియో
करना है। आज हम ऑनलाइन पर आ रहे हैं। अभी जो चर्चा हुई है कि ऑनलाइन जो है लगभग 100 से अधिक लगभग 100 विश्वविद्यालय ऑनलाइन के होंगे और अब जो आज सूची जारी हो रही है, जैसे कि यूजीसी ने अपने मानकों में तय किया है, यूजीसी ने मुझे अवगत कराया चेयरमैन साहब ने, और पीछे जो बैठक हुई थी कि जो नए का 3.26 है वो, और जो 100 टॉप 100 में जो भी ये विश्वविद्यालय आएंगे, संस्था आएंगे, वो स्वतः स्फूर्त जो ऑनलाइन शिक्षा है उसमें काम करना शुरू कर देंगे। कि बहुत बड़ी बात है, क्योंकि ये अवसर है, इस अवसर पर हम हाथ से गंवाना नहीं चाहते हैं, जाने भी नहीं देना चाहते हैं। हमारे सामने चुनौती है, चुनौती का मुकाबला हमको अवसरों में तब्दील करना है। तो मेरा भरोसा है कि आज जो ये उत्सव है हमारे लिए, जो संस्थाएं इस सूची में आई हैं, उनको मैं बहुत बहुत बधाई देना चाहता हूं, और जो अभी सम्मिलित हुए हैं, नहीं आए हैं, या जो सम्मिलित हुए ही नहीं है, मैं उनको शुभकामना देना चाहता हूं कि वो इसको आगे आकर के आगे आए और आगे अपने इस विजन के साथ कि हाँ वो शिक्षा में गुणवत्ता
मेहनत के लिए तुम्ही सगळ्यांनी
शिक्षा देने के लिए। तो तो सब
बघितलं होतं ना?
प्रमाण तो देना पड़ेगा हमको और मुझे भरोसा है
गुरुजींनी
कि
पण
Ravi Shankar, shall we start?
Yeah, yes, sir. We'll start, sir.
A warm welcome to respected speaker of the today's webinar and dear participants for the fourth day of online 1 week international faculty development program and skill development program on emerging innovations and insights in pharmaceutical sciences, jointly organized by Jawaharlal Nehru Technological University, Kakinada, and VV Institute of Pharmaceutical Sciences, Gudlavalleru, Krishna District, Andhra Pradesh. And today, Dr. Hari Saripali, popularly call it as a Harris. He is the research scientist and manager, Agri Testing Laboratories, a division of King Honey Limited, Topo, New Zealand, and he will deliver a lecture in the area of pharmaceutical biotechnology with a talk on biotechnology in drug discovery and clinical applications. Thank you very much, sir, for your patronage with our institute. And just I will give you a brief outline about the today's topic. Nowadays, biotechnology and drug discovery technologies, these are facilitating the rapid expansion on the clinical drug chest and empowering the clinicians with a better understanding of diseases as well as novel modalities for treating the patients. Important research tools and themes in pharmaceutical biotechnology includes tissue engineering, stem cell technology, genomics, proteomics, ligand receptor interactions, signal transduction, rational drug design, biochips, and microarrays. The emerging drug classes includes monoclonal antibodies, cancer vaccines, gene therapy, antisense strands, enzymes, and proteins. And during this COVID condition, the biotechnology is also playing a pivotal role for the treatment of the COVID-related aspects also. Today's, I'm happy to inform that our speaker, Dr. Hari Saripali, Harris, is having a sound knowledge and expertise in pharmaceutical biotechnology, and he is also a good researcher also. Now, I hand over the session to my colleague, Dr. P. Ravisha, coordinator for the today's webinar series, to introduce the today's speaker to the participants. Over to Ravisha garu.
A very good afternoon to one and all. Let us start the fourth day session of 1 week online international faculty development program and skill development program. Before going to the session, let me introduce today's speaker, Dr. Hari Saripali. He is the research scientist and manager, Agri Testing Laboratories, a division of King Honey Limited, Taupo, New Zealand. He mastered in microbiology. His specialization is In MPhil is botany, phytomedicine. He did his PhD in biotechnology, phytomedicine. His area of specialization is marketing management, information technology, and medical sociology. He supervised 5 MPhil and 2 PhD dissertations. His publications are 52 numbered in peer-reviewed national and international journals. He presented 40 papers in national and international seminars and conferences in India as well as abroad. He presented 40 papers in national he wrote 12 numbered textbooks, authored, co-authored, and revised editions too. He gave 14 numbered guest lectures. Now I hand over to the speaker for his session. Hand overing to the speaker.
Yeah. Hello.
Sir, please start.
Hi, guys.
Yeah, you can start sharing your screen, sir.
One moment, please.
Yeah.
Is it going? Hello?
No, sir.
Hello,
No,
sir.
sir.
Tell me. No, sir.
Yeah, now it's sir.
Yes, sir.
Now it's on, sir.
Please enlarge the screen, sir.
Yeah. One moment, please. Right.
Yes, sir.
All good?
Excellent.
Fine, sir.
You start the presentation, sir.
All right. Good afternoon to you all, but here is night, but okay, it's all right. First of all, I'm so thankful for the organization for arranging this faculty improvement program and invited me as a guest lecturer for today's session. Good afternoon to you all. Today I am going to give a lecture on biotechnology in drug discovery and clinical applications. And the tagline for this topic is Impact of Biotechnology in Pharmaceutical Technology. So already I think they introduced me quite well. I am just skipping this one. Yeah. And I'm working in a IANZ accredited laboratory. It is an international accreditation of New Zealand. And we are routinely doing testing of various samples, food samples, water samples, and other samples as well. And yeah, so my topic today is biotechnology in drug discovery and clinical applications. Yeah, and before going into the topic, so this is about me. I'm working as a research scientist and a lot of research papers in various journals, peer-reviewed international and national journals, and also gave a lot of lectures in various countries, in Africa, Australia, New Zealand, yeah, and India as well. Yeah, this is about me. I don't want to waste more time here. And coming to here, this is the introduction of our topic. So everybody knows, I may heard about the biotechnology. So what is biotechnology? So biotechnology refers to application of engineered principles in the field of biology to produce valuable products using microorganisms. So it is very easy to understand this concept. I'm not only focusing on the people who know about the biotechnology. So most of them involved in this session know something about biotechnology. So it is an application of engineered principles in the field of biology to produce valuable products using microorganisms. It's a very pretty easy. Or in other sense, we may also call it as application of biological systems, living organisms, and we can say it as either plants or animals or microbes, etc., or their derivatives in making of modifying products or processes for specific use. So that's another definition. So biotechnology is used in various fields such as agriculture, food science, pharmaceuticals. Coming to the pharmaceutical companies, why the pharmaceutical companies use this biotechnology? If this area of science having some sort of a benefits, what are those benefits? We would like to know. The pharmaceutical companies mainly are using this biotechnology principles or biotechnological methods manufacturing drugs. So most of you guys are pharmacy, you may know what is drug or what is antibiotic or whatever. It's sort of a any substance which has a physical effect when it is ingested, taking into our body or into the system. If it is entered into the health system, it shows some sort of a physiological effect. That's called as a drug. So manufacturing drugs and pharmacogenomics, and we will discuss more about pharmacogenomics in the coming slides, please. And gene therapy and genetic testing, the testing of various, the genomes depending upon the type of, disease. Nowadays, yeah. So the molecular science is evolving. So that's why we can study the genetic material, how the DNA molecule or whatever. See, to identify the defective gene, something like that, it's related with the genetic testing. So these are the things like building up or developing some genome, like DNA-based kits or molecular kits, diagnostic kits, or etc. Biotech companies make biotechnology products, specifically said biotech pharmaceutical products, by manipulating or modifying organisms. So usually at a molecular level and widely used in prevention, diagnosis, or treatment of many types of diseases. See, what is the purpose of manipulation? What is the purpose of modifying the organism? Okay, for example, to prepare a vaccine. Okay, so we have to take the organism and completely eliminate the pathogenicity of the organism without affecting the immunogenicity. So thereby, if we put that organism in the body, that organism doesn't show any pathogenic symptoms, any pathogenic effects. At the same time, the immune system triggers and releases antibodies in the body. So in such a, for that, there is a purpose, there is a target based on that target, we had to manipulate the organism at a molecular level. So that's called as a biotech. Biotech, especially the pharmaceutical products which are made by this one using biotechnology. Yeah. Today we can spot the historic milestones, it's nothing but called as the developments, achievements in the pharmaceutical industry. So a brief timeline is there. I would like to explain about that in the coming slides, please. If we see, for example, the era, like a golden period of era, something like that, even if you see that the current scenario, like 2020, is fully occupied with the COVID-19 virus. So something like that. Of course, whether is it good or bad, yeah. Always the people think in terms of a bad, but from the bad also, if we have a keen thought, learn something good out of it. That's called as, it's everything is depending upon our, what is called as perspective, the way we have to see the things. So in such a way, if you see the year 2003, that represents the 50th anniversary of discovery of double helical structure called as the DNA molecule. So in our body, there are so many organs and so many tissues, and each cell is made up, each tissue, each organ is, everything is made up of a basic unit of a human body or any animal body. The basic unit is cell. The cell consists a dynamic part that's called as a DNA molecule or the genome. So what is meant by gene? Gene is nothing but called as a segment of DNA. So that DNA molecule, so why that 2003 represents, it's a 50th anniversary of the discovery of the double helical structure of the DNA molecule. Whereas there is another great discovery that's called as rDNA technology. So that's the 30th anniversary. That year represents a 30th anniversary of discovery of the technique, rDNA technology, recombinant DNA technology. And this rDNA technology leads to the development of new recombinants and therapeutic proteins. Okay, what is meant by new recombinant? Okay, I think most of them you may visit or you may see in the YouTube videos how the people, how the scientists, how the research people are, they are making a new combination of different types of like hybridoma or etc. So the combining the two genes from different sources together, it forms a completely a new recombinant. Okay, so I'm telling you guys, there is a positivity, there is a negativity. Always, yeah. The pros and cons are side by side. For example, if you take two animals for your easy understanding, I don't want to go in deep that at the molecular level, take the X gene and combine with the Y gene, and then you will get a Z gene, something like that. I don't want to tell like that. Instead of that, for your better understanding, I would like to give an example. Okay, if I take some, the elephant is a big animal, the gene from the elephant, and I would like to take one gene from some reptile that's called as a chameleon, something like that. If I fuse these two, the gene from chameleon, it's a reptilian, it's almost look like a dinosaur, the small dinosaur, and in Telugu it is called as a tonda or something like that, or Vosaravelli, whatever it may be. We have to take that one, a gene from that, and one gene from elephant. My intention is I want to generate or create a dinosaur. I want to create a dinosaur. So the features, the gene which is responsible, I'm expecting that the features should be like chameleon. And here the size, the massive size is from elephant. So that's why I'm fusing these two genes, chameleon and elephant together. I'm expecting a massive reptilian feature. It's called as a dinosaur. So that's my assumption. So that's why I'm just doing this type of research But I don't know at the end of the day what will come. I may get a small size elephant. So the technology is always unpredictable before the emerging the biotechnology. Once after emerging this biotechnology, there is no possibility of getting this type of adverse things. Okay, we can try to, it's called as a, I think you guys may heard about this, called as a gene knockout and other science, a gene silencing. Okay, we can predict what is going on. Using the another science in combination with the biotechnology and information technology is nothing but called as a bioinformatics. I don't want to take much time on this slide. So new recombinants and therapeutic proteins. And coming to the another important project that's called as a human genome project is a very biggest milestone in biotechnology, leading to new genomic based drugs. But I'm telling you guys, in future, definitely, the drugs are not simply available nowadays. Everyone is going, if you're getting some pain in so and so part of your body, simply we may go to a pharmacy and getting some sort of a painkiller. But in future, the drug delivery system is completely going to be a novel or a different. A person-based pharmaceutical treatment, the person-based drug treatment will come up. Okay, take some sort of a blood sample from that individual and see the things, how the things are going on based on his age, based on his genetic genomic sequence and everything. They formulate a separate drug instantaneously and will give to the patient or the person or the people or individual. Yeah. So in such a way, the recombinant proteins or the genomic based drugs are evolving soon. So in fact, the pharmaceutical biotechnology is one of the key industry today. It's really become a key industry. I'll explain in detail to you later slides, in coming slides, please. So a brief timeline in pharmaceutical biotechnology. So in the year 1797, Edward Jenner is a Jennerian vaccination. He inoculates some viral vaccine into a child and protect that child from smallpox. If you see this history, this is quite interesting, it's amazing. Why it is amazing means Jenner, he did not know what is meant virus. Even he may not heard the word virus, but he invented a vaccine against the smallpox disease. How he invented? See, currently, we know pretty well more about the coronavirus. Everything like the shape and size and even the genome molecule, what are the spikes, what type of spikes on the virus, but still it is very hard to develop the vaccine for that. Still, yeah, the people are trying and yeah, the vaccines are coming up and still it will take some time, but I can say the coronavirus vaccine is one of the first, means the vaccine which is prepared within a short span of time. Definitely, I can say that. But see, we know everything. See, for example, we know the HIV virus, but still the vaccine is not available for that. How the Jenner did this without knowing the definition of virus? So we may all know, or some may know, some may not know. What is virus? Virus is an obligate intracellular parasite. Jenner discovered this vaccine based on the keen observation. If you need more information on this, yeah, I'm very happy. You can, flick a mail or just, take my contacts from Dr. Organizer of this event. Yeah, we'll provide a lot of details on this topic. I don't want to, just skip on to the next one. So the Patcher proposes microbe cause fermentation, and penicillin discovered by Alexander Fleming. It's called as an antibiotic. You know, guys, what is antibiotic? Produced from one organism, used to kill another organism. It's called as antibiotic. So obtained from one organism, used to kill another organisms. Right. And in the year 1944, Avery proposes DNA as a genetic material. Next, Waxman isolates a streptomycin antibiotic for the disease tuberculosis. And yeah, 1953 DNA structure, and 1967 protein sequencer, and 1970 discovery of restriction enzymes. These ones play a very important role in recombination DNA technology. And 1973, the first recombinant DNA in bacteria with restriction enzymes and ligases. 1977, human protein in bacteria. 1980, patent for gene cloning. And 1981, first transgenic animal. You guys know what is transgenic animal? Yeah. And in the year 1982, Humulin, the first recombinant biotech drug approved by FDA, Food and Drug Administration. And 1983, invention of PCR, polymerase chain reaction. So this is the method currently. The means all the the diagnostic are the people who are providing the service in the labs and investigating whether the person is either COVID positive or COVID negative using this technique, polymerase chain reaction. And in 1986, the first recombinant vaccine for hepatitis B. So hepatitis is inflammation of hepatocytes. It's nothing but called as a liver. That's the first vaccine, recombinant vaccine. And 1988, the US patent for genetically modified mouse. That's called as oncomouse. In 1990, launching of the first human genome project and first somatic gene therapy to cure ADA, SCID. It's called as a severe combined immunodeficiency disorder. We can see this type of condition, especially in case of the people who are suffered with immunodeficiency disorders. Even we can see this type of situation, especially in case of the HIV patients too, because their immune system is very, very weak. So in that individuals, they built up the immune system by using this somatic gene therapy. The next, transgenic cow produced human proteins in milk. In 1994, the DNA is for cystic fibrosis. In 1997, the first animal cloned from adult cell, that's called as a dolly. This is called a gene cloning animal. This is completely against the, we may say, nature. Usually in the nature, the formation of an individual is possible through a combination of male cell and female cell. But in case of this dolly, 1997, the dolly, the first, gene cloned animal. Okay, so it means there are so many other animals also there, but this experiment was successful. If the experiment is successful, then only we have to speak about that. Otherwise, yeah. If it is failed, the first one still has to come. So the dolly, the first gene cloning animal, is synthesized by using two female cells, one embryonic cell and one cell from the same animal, udder cell. And that udder cell subjected for electroporation, lot of electrical shocks, and thereby the somatic genes, means the sex chromosomes in that, might activate it, and that female cell act as a male cell, and these two cells are combined together to form a dolly. And in the year 2000, rough draft for human genome is announced. It's not completely finished, but rough draft. In 2002, draft version of the complete map of human genome is published completely. And first oligonucleotide drug is approved by Food and Drug Administration. It's like an oligonucleotide molecule. Next one. RDNA technologies entered into the drug discovery and all fields in the development and manufacturing of therapeutic proteins and nucleotides. So BT, biotechnology, has a major impact on pharmaceutical industry because of recent advancements in recombinant protein chemistry, vaccine production, and diagnostics. Treatment of many serious unmet diseases. There are different types of diseases. Some of the diseases is very hard to find out. Even such diseases are also treated by using this biotechnology. Currently, approximately 150 approved therapeutic proteins and vaccines are available. Recently, already I explained to you, the first oligonucleotide. Is it oligo or oligo? Yeah, the people of, even my teacher, I remember still my teacher, he used to call it as oligosaccharides or oligonucleotides, something like that. So oligo means a short chain nucleotide for the treatment of cytomegalovirus infection. So this cytomegalovirus is responsible to cause an infection for the eyes. So it is called as a retinitis. Retinitis means inflammation of the retina. So this drug first approved by Food and Drug Administration. The drug name is called as a fomiversin or vitravin, is an antisense oligonucleotide and represent a new biotechnological group of compounds with new promising therapeutic purposes, especially for immunocompromised people. So already we discussed that. What is immunocompromised one? Sorry guys, my screen is completely occupied with the bits and pieces of all this stuff. That's why I'm taking a little bit time. Yeah, pharmaceuticals and biotechnology. Benefits of combination. So what is the reason of fusion or amalgamation of different areas? So this is a very, I can say, yeah. So there is a more market. So why the people are doing a combination or amalgamation of the things? Because of seeing more benefits. Okay. So I'm just going for a few seconds out of the scope. Okay, take an example of the movie called as RRR or whatever. Yeah, there are two people. They're combining means one. Yeah. So there are two heroes. Their fuse means combination of these two. The reason why, because they want to capture more market. So I can say here why they're combining this pharmaceutical sciences or pharmaceutical technology and biotechnology. What are these benefits? When these two disciplines, pharmaceuticals and biotechnology, come together, the result in many advantages for mankind in terms of healthcare. This is not in terms of business, but I will talk about the business later on, because yeah, without economy, without money, or without what is called as a profits, or it's very hard to manage anything. So it's a highly necessary also and essential also. I will explain that one later. So because of many advantages, what are those advantages? I would like to explain in the coming slides, please. This is possible through pharmacogenomics. This is possible through pharmacogenomics. So whatever those advantages related with the healthcare, they are possible through pharmacogenomics. So it is derived from the two words again, that's called as a pharmacology and genomics, which refers to the study of how the genetic inheritance affect the individual human body response to the drugs. This is a very important thing. I already told to you guys. Every person is different. Every person is different. I can say that you are unique in this world. You may see so many people around you. Okay, all are human beings only, but you are only one piece in this entire globe. You are a unique one. Similarly, your body system is different. Your immune system is different. It is not a common factor, even though the organs are there. But how the cell is responding towards some factor, we don't know. That is only the reason called as the study how the genetic interference affect the individual human body response to drugs. So biopharmaceutical drugs aim at designing and producing drugs that are adapted to each person's genetic map. This is a very important thing. Your DNA molecule, of course, the nucleotide sequences, nitrogen bases are the same, adenine, guanine, thymine, cytosine, but their arrangement is completely different. So based on that, yeah, your health system or the physiology functions, everything is going on. Pharmaceutical biotechnology benefits of combination. See, pharmaceutical biotechnology companies may develop tailor-made medicines for maximum therapeutic effects. What are tailor-made medicines? This is quite interesting. That's called as earlier days, the technology is not that much evolved. If the people are suffering with something, okay, they will take an example of, I don't want to mention the name of, any healthcare system, because all healthcare systems have their own importance. Take an example of, okay, in general word called as herbal medicine. Okay, the earlier days, the ancient days, the people go, if they collect some sort of a plant and they will, they will grind it, make it as a paste or something like that, powder, and combine with honey or some other materials and put that one wholesome into the body. If we eat around, I don't know, so many grams of food, that material, leafy material into the body, we may get the enough amount of, means it may consist only the traces of medicine, particular medicine. So the remaining everything is unnecessary to the system. And similarly, even after that, the vaccine, okay, developed at the early stages. Okay, if they put that vaccine there, okay, I think there is a lot of scar. It's a big scar, and the people are suffered with a lot of, vomitings and even the body temperature, like so-called as side effects. A huge side effects, because they are putting with that immunity generating agent. In addition to that, they're also putting unnecessary stuff into the body. That is the reason getting some side effects. If a tailor-made medicine means exactly which part of that entire organism will act as a, see, for example, if you see a pathogen, the pathogen may consist the two important factors. One is immunogenicity, another one antigenicity. That antigenicity is may responsible for causing the pathogenic disease symptom. Immunogenicity means the ability of producing immune system. So only that particular segment isolate from that organism and insert into the body as a therapeutic drug or the protein. So thereby it will obtain a maximum therapeutic effect. So will benefit quite a lot. So that is the use of the benefit, the tailor-made medicines using this biotechnology and pharmaceutical. The major benefit of pharmaceutical biotechnology is in the form of better vaccines. You know what is meant by vaccine. So everybody heard about what is vaccine, but I would like to define the vaccine. I don't want to give a big definition. It's for easy understanding. It's inactivated microbial suspension. How you inactivate, either killed by or the live attenuated, whatever it may be. So depending upon the nature of the, or it's a recombinant vaccine, okay, the method which we had to employ to inactivate the organism and without affecting the immunogenicity, if insert such a molecule into the body, so our immune system recognize that one as a immunogen and produce antibodies and develop the resistant power against the disease. That's called as better vaccine. Biotech companies design and produce safer vaccines by using organisms that are transformed through genetic engineering. Okay, you may use either E. coli or adenovirus. In terms of coronavirus, they're using a vehicle that's called as adenovirus. And they're taking that particular portion from the coronavirus and inserting into the adenovirus, and they form a vaccine. Otherwise, it may not go into the body, it may not it may not generate the antibodies what we are expecting. So these biotech vaccines minimize the risk of infection. This is a very important stuff. So the purpose of intake of vaccine is to develop the immunity without causing any infection. But we may heard about some sort of stories that the polio vaccine, the people got the polio after that, because this is, see, there are so many things, not only the preparation, after preparation also, the proper preservation of that vaccine is very important in the health centers. And proper administration of that vaccine into the body, that's also another thing, proper inoculation into the body. So if they are not supposed to do in a proper way, yeah, the infection, the risks are, the side effects are more. And coming to biotechnology and its applications in pharmaceutical industry. I'm not talking about any technique here, because if you open, the Google or YouTube video, you will find out a lot of techniques. Like if you type a gene therapy, yes, you will get some sort of a nice video with animated video. So everything will come up and you'll know what is that. If you're really interested in biotechnology methods, yeah, you can do in such a way to learn more about that. But my focus is here, biotechnology and its applications in pharmaceutical industry in terms of industrial scenario, industrial aspects, please. More than 150 approved biotech drugs or vaccines are on the market now. 150 approved drugs. And 70% of biotech products were approved in the last 6 years, 70%. So the approvals, if you see the approvals of the new drugs, 70% of the drugs are approved. As per Pharmaceutical Research and Manufacturers of America, Pharma, so they did some survey and they identified that 369 drugs are in pipeline, meeting the criteria as a biotechnology drugs and medicines. So they did some sort of a survey and finding out how many drugs are in the pipeline, means the work is still going on. These drugs mainly target the potential diseases. Okay, friends, I would like to explain here what is the difference between the infection and the drug. In the previous slide, we discussed about the infection. Minimize the risk of infection. That's called as a better vaccines. Here called as the diseases. What is disease? The initial step of disease is called infection. The infection progresses turn into a disease. So the drug are these drugs which are in the pipeline, they target the potential diseases and they provide a new therapies for some sort of disease like autoimmune diseases. I think you might know what are autoimmune diseases. Autoimmune diseases means this disease is not coming from the outside. Even in our body, the cells, our immune system recognize our own cell as an antigen and produces antibody unnecessarily in the body. Filling our body completely with the unnecessary antibodies are unnecessary WBC cells because they are not required. So that leads to causing some sort of, I think you may heard about called as a hypersensitivity. I don't want to go in deep in that. So hypersensitivity is nothing but called as an allergic reaction. Exaggerated immune response. Exaggerated immune response. So those means like an formation of immune complexes and other, anaphylactic reactions. Okay, those are called allergic reactions. These autoimmune diseases, mostly predominantly, we may say Parkinson's disease, Alzheimer's disease, something like that, called as autoimmune diseases. Asthma is called as a bronchitis, unable to take the breath, and also there are some people have some sort of genetically they got the asthma. And Alzheimer's, multiple sclerosis is called as a cancer. We may also call it as a bone marrow cancer, and including immunization and different infectious diseases like even AIDS also. So there are different types. Biotechnology produces pharmaceuticals currently account 5% of the worldwide. 5% of the worldwide pharmaceutical market and are expected to reach approximately 15% by the year 2050. So it is going on progressing. It's going on progressing. So by the year 2050, it will reach to 15%. Sorry. The explosive growth of genetic diagnostic techniques will allow personalized genetic profiling of each individual in 1 hour for less than $100. See how much they are spending, how much they're, the explosive growth of genetic diagnostic techniques. So those techniques are completely related with the individual base. It's a personalized genetic profiling. Pharmaceutical biotech industry producing hundreds of diagnostic tests, reducing cost of clinical analysis. So for everything, there is a, different types of tests. Okay, for 3 tests, 3 times the patient has to go and give the blood sample to do 3 tests, something like that. So this is a very easy one. So there is a lot of cost. There is a lot of what is called as the time to do that one. We need a lot of manpower to do all this stuff. But using this biotech diagnostic methods and kits are developed using this biotechnology principles and methods. It's very, for example, take an example of here, testing of low density lipids, lipoproteins, or lipids, or cholesterol and other parameters in one test design in comparison to conventional tests like cholesterol, total glycerol, LDL or determined separately at higher cost. So it's like a technology-based one and conventional based methods. The conventional is a time-consuming and also cost-expensive one, whereas these technology-based techniques or methods are a very simple one. Within short time, the rapidly will get the results. At the same time, it's a cost-effective thing. In future, biotechnology-derived tests will be more accurate, quicker than the previous tests, will allow earlier diagnosis of diseases. So this is also very important. Take an example of cancer. If we diagnose the cancer in early stage, in the stage 1, it's very easy to control or means we can extend the life of the person. But it's very hard to find out in early stages. So by using this biotechnology, we can find that. And coming to here, the proteomics, useful to discover the unknown molecular markers. Those are responsible for causing the diseases before the disease symptoms appear. So helping to prevent the disease and even conduct therapy is much easier. And xenotransplantation. This is from a transgenic animal. It's a future field in pharmaceutical industry. So it's nothing but called as, you guys may have heard about the organ transplantation. So the organ transplantation is an effective and cost-efficient treatment for severe and life-threatening diseases of organs, mostly heart, liver, kidney. These are the things. See, if you see the recipient list, the organ recipient means the receivers, the required, the people, how many people are required these organs in different countries. In Europe, around 35,000 people are registered for the transplantation purpose. Whereas in the United States, there are around 60,000 people. This is not the recent one. This is, I think, 4 or 5 years data. But if we see the current data, definitely there are so many number of people or the recipients for the organs. And apart from the, there is another method that's called as a tissue engineering. It's an attractive field in pharmaceutical biotechnology. So this tissue engineering combines advances in cell biology and biomaterial science. This is a very important area as well. Tissues consist of scaffolding material, like it's nothing but called as a collagen or biodegradable polymers or some other things, which eventually degrade after forming the organs or cell implants. Skin tissues and cartilages were the first tissue successfully engineered. Recently, this biohybrid systems to maintain patients' liver or kidney function were also successfully tested. See here, the transgenic animals, if we are getting the organ from another animal, sometimes the system, the immune system may not supposed to receive that organ in a proper manner because of, non-homologous nature of these organs. The tissue engineering means the same. We had to take the cells from the person and we have to, make like a engineer that entire organ in the artificial environment. So thereby the acceptability, the percentage of acceptability, the compatibility is more compared with that of the organs generated by, see, the technology. For example, the people may thought that, yes, so already this is there and this technology have some disadvantages. Yes, if the technology, the current existing technology have some disadvantages, then there is a growth in that particular sector. Imagine if there is an Android phone with full of applications and everything in that, there is no, it's not possible even to point a single disadvantage. Then there is no growth in that item. There is no growth in that product at all. That is the saturation point of that particular product for particular area. So if you see guys here, every technique, every method have some advantages and disadvantages. If you identify, see the disadvantages and try to overcome that one, and then the new thing, the new method will evolve. The new technique will evolve. But if you say that the existing one is not good, so when do you know it is not good? If you use that one, then only you will know that it is not good. If you study that one, then only you will understand that that one is not good. So then, yes, you take up and try to overcome, try to solve and give the better one for the community. So biohybrid systems is the better one compared with that of the organ transplantation system. And the next one, please. Stem cell technology is a new avenue in therapy to cure most deadly and diseases such as even the Parkinson's, Alzheimer's, leukemia, cancer, genetic disorders, deficiencies, and cystic fibrosis. So there are different types of diseases are treated or controlled, treated by stem cell technology. What is the impact of biotechnology and genomics on the drug development process? The most frequent trends for the pharmaceutical industry in biotechnology, yes, surely new technologies and innovations, especially genomics. Already we discussed it in the previous slide called as pharmacogenomics. And apart from genomics, also influences of government regulations, healthcare legislation affecting own product pricing, changes in demographics, and aging population. There are so many factors. If you see that, there are some developed countries, there are a lot of genetically modified food or modified plants are available. So the people are accepting the technology. But in some countries, they are not because of, there are so many factors. So considering all the factors like government regulations, healthcare legislations, everything has to take into the consideration to release a new drug. With special emphasis, more topics with minor influence related to, see, apart from genomics and other major topics, there are some special emphasis, minor influence related to patent production. Whatever you are getting, you have to try to get the patent, otherwise it's very hard. There are so many people, they photocopy that one or just copy that one like a capricorns, and then you may lose, to make one drug, there is a huge investment. We will discuss that one in the coming topics, please. So multinational scope of industry. So whatever the industry, your goal should be a multinational. Okay, so I will make some sort of a drug. Okay, I will use in this village or use for this government, and I'm focused only the government of India. I don't think about the other areas now. Whenever you have to, you try to widen yourself. Requirements for new drugs. So it's always there, because whenever the new enemy means a new organism is coming, infectious organism is coming, yes, yeah, we need a new drug to control it. And changes in information technology. So this, the globalization, the changes in information. Okay, whatever the information, the cloud system, cloud-based system, there are so many things. Yeah, in future, we can say that, yeah, we may not see even e-pharmacy. You guys may know that e-marketing, web marketing, social marketing in such a manner, we may, in future, we may see e-pharmacy shops, outlets. So in the first decade of this century, the biotech industry is likely even more explosive growth progress in computer modeling, like a CAD, if you see the CAD, computer-aided drug designing, something like that, automated lab techniques. If you see the laboratories, there are so many robots are there. They will do the technique, in a faster manner. You take ELISA readers or whatever, so many heaps of samples, even 300 or 500 samples at a time, they will process and they'll give the results. You see, the Middle East, they are really doing an amazing COVID testing in Middle East. If you open and see the Dubai and Saudi, how they are handling these COVID situations. So that I can say that's the first and, the first country doing heaps of tests, some thousands of tests every day within a short time. See, once the sample is given to that people and you don't even go for the results are directly come to your mobile within a 24 hours of time. See, this is all called as automated lab techniques. So knowledge of human genes and protein continues. As a result of more life-saving therapies will reach the people. Today, pharma industry seems to be a good shape to work. Sorry. Yeah, today pharmacy seems to be a good shape to work on these challenges. Some impressive statistics of industry growth. Would like to see those statistics of the industry growth. During 1980, the biotech industry turned out 80 new drugs and vaccines. 1980. By comparison, 33 biotech medicines were approved in both 1998 and 99. 25 more were approved in the first half of 2000. the number of patents granted to biotech companies has tripled from nearly 3,000 per year in the early 1990s to more than 9,000 in 1990. See, I would like to, this is not like a number game, something like that, but I would like to tell that how this area is progressing. I would like to share with you that thing. You know, if I share this is progressing, you may not, okay, so how it is progressing? By seeing the numbers, then you'll understand, yes, this area is really progressing good. Okay, after a decade of slow, steady growth, biotechnology patent awards began a step, and it's growing really logarithmically. See, in 1995, nearly 4,000 patents were granted. 4,000 patents. Then a number of patents skyrocketed at a rate of 25% for more each year. The pharmaceutical companies would usually see, traditionally, they have focused on chemical approach to treating disease, have become increasingly supportive of biotech and R&D. Slowly, if they're capable, they may establish their own labs, or otherwise they're partnering with other biotech firms and biotech organizations and coming together, you know, a part of the work, do the basic work there, and then the biotech related work. If they have their own lab, they can do that one. Otherwise, they can give that work to the other, you know, the biotech associations. They will perform on behalf of them, like a mutual understanding or agreement. Between 1998 and 99, the industry-wide sales and revenues increased by 13%, means 16.1 billion in 1998, whereas in 1999, 22.3 billion. See how the tremendous growth. For the future, the biotechnology and genomics, pharmacogenomics, or biotechnology and genomic way, technology-driven and formed by integration of high throughput technologies, genomics. Yeah, already we discussed about bioinformatics. Yeah, so even the genetics wave is a data-driven and is applied new life science field to identify genes that make individuals as their careers susceptible to a particular disease, and allows a personalized medicines based on pharmacogenetic facts, based on their pharmacogenetic facts. So that's why it is a very important and high throughput technology. Impact of pharmaceutical biotechnology and genomics on the economics of R&D. So why this area is really good enough. The first one is reducing cost in R&D. If using a conventional methods, there is a lot of investment. Using with, I will explain with this example, then you guys will understand very clearly. So I'm about to finish within 5 to 10 minutes, guys. Please, thank you for your patience. Reducing cost in R&D. Before biotech have been extensively introduced to industrial research, developing cost of each drug has had cost companies on average $880. That's $1 million. $880 million, and had taken 15 years from start to market authorization. You know, the clinical trial 1, 2, 3, and final approval. So about 70%, 75% of this cost were spent on failures. This is a very keen and interesting thing. So whatever the investment they are invested for the development of a new drug, 75% of that investment is simply a failure. Using genomic technology. So I can't say that, every research is definitely going to give a good results. Yeah, there is a chance of getting the false also. We have to accept that. But coming to here, the probability will increase if we're using genomics. There is a realistic chance of reducing companies' cost. A result of efficacy gain significant savings, not only related with the money, but also with the time. See, in industry, especially in industries, I'm telling you, time is money. The people may thought that the money is a capital investment, something like that. The time is also money. So if you finish within a short span of the time, that is only the reason why the rapid techniques are coming, evolving every day by day. Rapid, the PCR, just like to finish within 10 minutes or 5 minutes. So such a means the PCR ready-made mix is also available. You don't need to mix, or we don't need to make all the solvents. So everything is available like a PCR mix. Okay, take that PCR mix like a COVID PCR mix and put it in that, the patient's sample and put it in that PCR machine, and immediately we will get the results. So that's called as the rapidity, means the time saving. The time, within short time, we try to get much more benefits. And another important factor here, increase in productivity. So now one is reducing cost, the second one increasing productivity. From trial and error approach to complex biochemical in vitro assays. This is a very important, and I like this one very much. Complex biochemical in vitro assays. So we had to design the same system outside the person, outside, not supposed to put that entire thing into that individual and we'll see whether is it working or not. Instead of putting in that experimental animal and see how it is working, instead of that, in vitro setup, and then do these complex biochemical things. And see, there are some sort of techniques like a microarray technology and single DNA chip. We can see the thousands of genes are diseased or even health issues will be analyzed by using a single DNA chip. So we don't need anything, any tissues from the body, just one blood sample or one particular tissue sample, one genomic sample, not even that. A fragment, what is gene? A fragment of DNA. Whole DNA is also not required. Just a fragment of DNA is required. And then we had to put that one in the PCR. So the single gene chip is responsible to provide all the information. By using bioinformatics, different assays can be analyzed and linked to integrated follow-up of information in databases. Yeah, there are so many examples of if the data is information is available. I think, guys, I will tell you here one important and interesting thing. You know that Amazon. Yeah, so that Amazon is a very, very, they started with, the book, selling books online, like a online book library. But you can see now, it's a very biggest company because they are selling anything and they're using a novel methods, novel technologies, anything. See, what is the driven? Nothing. They are doing some sort of, the customer, how the customer is, means thinking. So in the same manner, as a pharmacist, we have to think how we have to care about the people, how we have to design a new drug to treat different types of diseases. So your focus should be only on particular thing. So if we have such a data, and that database, like a bioinformatics, which is really useful for us. Like take an example of CAD. We can, synthesize the vaccine using in an artificial means, not in artificial, it's a dry lab. It's nothing but called as a dry lab, using different types of Schrodinger software, using computer-aided drug design, something like that. This is possible once the database is available. The database of the virus, the database of the drug, the database of the protein, the database of the ligand, and we had to fuse all these things together with a platform that's a Schrodinger platform or whatever the drug designing platform softwares. So see, for example, here the Boston Consulting Group calculated a 6-fold increase in productivity at the same level of investment. So by using this technique. And coming to here, accelerating the drug development process. Already I told to you this one. Okay, so this is nothing but called as the time, the preclinical development of a drug, biotechnology and genomics play a very important role to help to predict the drug properties and pharmacokinetic properties to accelerate the industrial drug development process. Companies, so that's why this is a very important part of the value chain. Okay, so the development process, accelerating development process is an important part of the value chain. The potential savings in order of 20 million, and see the drug 0.3 years, so very less time. And maintaining high standards in quality assurance, this is also very important. Okay, once we are getting one drug, and that drug should pass some sort of a method validations and then do some sort of a quality assurance check and gets released from the compliance and other stuff. So biotechnological drugs have the same high standard in quality and safety compared, as a conventional drugs. The main reason for this extra time needed for unknown, if the something is not known to us, then you have had to spend like much more time. And also importance of developing new drug specific appropriate test assays, also very important stuff. The common pharmaceutical biotechnology products that are made by the biotech pharmaceutical companies. So these are the basic things, and there are so many other stuffs are also there, mainly antibodies, proteins, and recombinant DNA products. So antibodies is nothing but, okay, it is a protein produced by the white blood cells and produced by the antigens. So again, as either bacteria or the viruses or the foreign substances, sometimes even I told you that regarding autoimmune diseases, there were self-antigens as well. So the monoclonal antibodies, the technique is called as a hybridoma technology. Okay, so they're fusing the different types of, myeloma cell and then the antibody cell and producing a hybridoma monoclonal antibodies. Coming to the proteins, they are made of amino acids, larger molecules. So that's called as a protein biotechnology. And emerging, one of the key technology for the future understanding and development of many diseases like cancer and amyloid formations or better therapeutic interventions in future. And recombinant DNA products. Okay, so recombinant DNA, deoxyribonucleic acid, is a genetically engineered DNA created by the recombinant fragments of the different sources, different organisms. So the products are vaccines, drugs, enzymes, growth hormones, insulins, proteins, and ACE. So biotechnology, the use of advances in life science in the production of, so these are called as, I'm discussing about the large companies. We would like to see the largest biotech companies in the world. Okay, so biotechnology, the use of biotechnology. Sorry, guys. So the main purpose of this one is in the production of marketable goods. This is a very important thing. Whatever the pharmacist or the researcher who decide whether that product has to be marketable. That's a very important thing. In terms of stability, in terms of efficacy, in terms of controlling the disease, treating the disease, or etc. So it is an enterprise that produces products, services by utilizing biotechnology. So I'm just speeding up here now. There are so many companies. These are the 20 companies, the highest companies. You can see here Johnson & Johnson, Pfizer, Roche, Novartis, Merck. And there are some more companies as well here. Yeah. So there are different types of largest companies in the world. Novo, Celgene, Bayer, Eli, Allergan, Astrogen, Biogen, Abbott, Share, Pharmaceuticals. So see how their market share, the value in the world. So how many billions? This is not the recent one. It's a most recent study things. This is the future look of the biotechnology, pharmaceutical biotechnology. So it is integrating biotechnology and genomics in the world drug development process, gives companies the opportunity to save the money, and also not only the money, even in terms of the time as well. Okay, each day, see, there are quite challenging. Any predictions for the new futures are challenging. A significant increase of recombinant drugs replacing up to 30% of commercially used low molecular drugs. If you see here, there are different types of areas in which the biotechnology and pharmaceutical biotechnology play a very important role. biological products, pharmaceutical preparations, diagnostic substances, cosmetic perfumes, etc., laboratories like automated lab systems, medical devices in terms of tissue engineering or organ transplantation, even like a phytomedicine, like medicinals and botanicals and other stuff here. So the production of recombinant biotech drugs, bioprocessing, and in all reactor sizes will be routinely, and gene farming, somatic gene therapy, we discussed all these ones, and biochip. But interestingly, creation of artificial life or complex biochemical networks expected to be unrealistic in the next 25 years. Thank you, guys, for your patient listening. Thank you so much. I'm taking a little bit more time. Sorry for this. I'm just, moving all this stuff while I'm seeing these screens. Yeah, this is called as a e-pharmacy. I told you guys. Yeah, any questions you can, you're welcome. Yeah, so thank you all. So this is my last slide. The revolving, the revolving earth. The engineers build the world, but the doctors save the world. The pharmacists care the world. Guys, I'm really thankful to all the pharmacists who are really helping quite a lot during this season, not only in India, even throughout the world. Thank you, guys.
Ravishagaru.
Yeah.
Over to Ravishagaru.
Sir, there is a question from the participant, sir.
Yes, please.
What is the current status of pharmacogenetics in primary healthcare? What is the current status of pharmacogenetics in primary healthcare?
Okay. Is there any other questions or only one?
Only one, sir.
Okay. Yeah. See, currently there are different countries, probably, yeah, noticeably, if we see the noticeably more than, yeah, every country is coming that, and they are doing some sort of a research. They're, they're producing the COVID vaccine. For example, I'm telling you, only this scenario, the current situation is like this. That's why I'm explaining that. See, this one is possible only because of this genomics, pharmacogenomics. Okay. If, imagine, there is no genomics at all. Taking that COVID virus or coronavirus, whatever it may be, how to trace, how to find out what part of that one is responsible to really causing some sort of infection in the body. For example, see, for infection, we need the primary, essential, the prerequisite of infection is only one thing. The organism has to come into the body, it has to attach. Attachment is the primary step. If anything, the antigen is coming into the body, and if it will touch, called as a receptor. The reason why, for example, I would like to give a clear example, then you'll understand very clearly. See, this receptor and antigen, what is called as absorbent, that absorbent and receptor, they are compatible, then only it will come and affect. Why the coronavirus is not coming and affecting the other organs, like, the eyes or the liver or other, why it coming and affecting only the lungs? Because the receptor was there in that. So these all studies are possible only through genomics. So we couldn't say that the current status means, yeah, it is very hard to say that, okay, only genomics is responsible to identify this one, or only the pharmacy can, like, okay, take an example of one medicine called as a malaria drug. It is useful to treat the coronavirus, something like that. Okay, so that pharmacy drug, how it is working, how it is going into the body, how the physiology is affecting, everything is related with the pharmacogenomics. So we couldn't say that the current status or the future status, something like that. Still, it is going. It is very effective. It is effectively going, but the people will not understand. But all the scientists, definitely they will understand. Even the pharmacists who knows about the pharmaceutical biotechnology, yes, the pharmacogenomics really play a very important role, especially designing a novel drugs within a short span of time. With not only that, the cost effective within short span of the time, it's play a very important role. Yeah.
Thank you, sir.
All good.
Thank you, sir, for your very nice session about the various concepts involved in biotechnology and their applications in pharmaceutical industry, along with the trade in very detailed and a very systemized manner. Thank you a lot, sir. And there is a special note to the participants. Please make sure that your name, designation and name of the institution must be properly entered while you are doing your feedback form to get your e-certificate in a very proper manner. And there is one more note, the feedback link will be shared in YouTube link description box too. I would like to hand over to the convener.
Thank you, Ravi Garu. First of all, let me thank Dr. Harris Garu for your excellent talk on biotechnology in drug discovery and clinical applications. Really, you got the excellent feedback from the participant audience, sir, from both Zoom as well as YouTube live coverage. Okay, and thank you very much for your excellent presentation. Really, the participants will gained a lot of knowledge on the biotechnology related to the pharmaceutical technology. And on behalf of VV Institute of Pharmaceutical Sciences, I would like to take this opportunity to express my gratitude and thanks to the today's speaker, Dr. Hari Saripali, and popularly call it as a Harris in the New Zealand. And my sincere gratitude to you, sir, for your excellent presentation for the our international webinar series. And I especially thank all the participants for the patient listening for this particular fourth day of this particular webinar series. And I also acknowledge and thankful to my colleague, Dr. P. Ravisha, and coordinator for the today's webinar series for her coordination in excellent manner. And a small announcement to all of you, on tomorrow, day 5 session starts at 9:30 a.m. The subject area for tomorrow's webinar series is pharmaceutics, and the speaker for tomorrow's session is Dr. Kamal Dua, lecturer, School of Pharmacy, Graduate School of Health, University of Technology, Sydney, Australia. He delivered a lecture on targeting inflammatory diseases using nano drug delivery. Thank you, one and all, and have a nice day. Thank you, sir. Thank you very much.
Thank you. Thank you. See you guys later.
Thank you, sir. Ravi, shall we wind up?
Yeah, yes, sir.
Okay, and thank you.